Today I'm going to be talking about TrueAllele investigation of DNA evidence. TrueAllele is a software that analyzes complex DNA evidence developed by Cybergenetics. Cybergenetics is a Pittsburgh-based bioinformation company that I founded over 25 years ago as a spin-off from Carnegie Mellon University and our work that we did there. We started off looking at genetic markers for disease diagnosis and drug discovery. And in 1998, the British government contacted us because they had a database problem. They had 350,000 profiles, and their staff was spending all their time at buildings filled with people, like 100 people, including administrators, just looking at the data. And they still had a 1 in 2,000 error rate. And we had automated genotyping for simple DNA in genetics, and so we just moved it over to forensics, and they were able to process 350,000 cheek swabs, not the laboratory processing, but the human review. The data review was automated by our computers. And so 100 people disappeared. They were left with 5 people, 2 desktop computers, and much better accuracy. And at that time, their required throughput under the law was either 3 or 5 days from beginning to end, from sample collection to getting samples onto the database. So we were able to take care of their time problem as well. And then they got us interested in the DNA mixture problem, which I solved 20 years ago by coming up with a way of looking at all possible genotypes that could go into a mixture, separating out 5-person, 10-person mixtures. But the idea was, instead of having people looking at limited amounts of data and limited numbers of possibilities, to have a statistical supercomputer consider every possible genotype of the trillion genotypes that could exist. Because it was everything, we had to put probabilities to that. We came up with a way of getting accurate probabilities by accounting for the random variation in PCR. And that, through our product and other products, is becoming the dominant approach to how to get more information out of the same data that laboratories generate. I could go on and on, but I want to start talking, and you'll hear more about this as we go. So let's begin. And what I wanted to do was to start with a case in Pennsylvania, in Lycoming County, because by having a case, it puts things in context and lets me go through what I showed the jury so you can learn here as students what it is that the computer does when it separates mixtures and analyzes low-level DNA. So that's what we're going to do for the first part of this talk. So here's this case, Commonwealth of Pennsylvania versus Dante, Washington. In 2014 in Williamsport, there was a cabbie who was shot by a passenger. He ended up surviving, but that wasn't clear at the time he was shot. So it was an almost homicide, but fortunately it wasn't. And there was evidence that was from which DNA was collected that involved the receipts from the taxi cab. There was also evidence from places on the car, touched DNA from outside handles, from the seat. And the question was the defendant present in the car? And could the DNA show that? And the state crime lab, Pennsylvania State Police, developed excellent DNA mixture data from these collected items. And they did the usual manual DNA interpretation, and they found that the victim, Mr. Phillips, his DNA was on the cab voucher and a zippered pouch, but, and this is quoting from what crime labs around the country report in very similar language, due to an insufficient quantity of DNA and the complexity of the mixtures, no interpretable results were obtained. Have you ever seen that language? Okay, so I'm telling you what you do when you get that language. That's what this talk is about. Okay? And in particular, that was for a rear right door handle and the rear right seat of the taxi. So there's no information, but if you had information that involved the defendant maybe together with the victim in a mixture. Mixtures are really powerful. They can place 2 people on the same item if you can resolve those mixtures. And so they were sort of stuck. So anybody know Ken Maines? He used to be a cold case investigator. He's done many things. Anyway, he was working from Lycoming County, and he had gathered the data to his office from the Pennsylvania State Police crime lab in Greensburg, and that's how you would get data. You would ask the lab to give it to you or to a prosecutor, and then he asked us to reprocess the data. So that, think of it as two parts. Evidence comes into a lab. The lab transforms the evidence items by extracting DNA, which you just heard about, and amplifying it with PCR, and separating it on the DNA sequencer to generate data. The output are data files. And these are the data files that can either be interpreted or not. And if they are interpreted by human review or simple methods, great. Otherwise, those files could be sent on for more sophisticated processing. And so he sent them to Cybergenetics, where we, at his request, we conducted a free data screening. In any case that Cybergenetics is involved in, we ask for the data first, and we will do a free quick look with the TrueAllele computer. This is helpful if you happen to work for government. Free is a good word. It gets rid of a lot of time and paperwork. Some of the work we do is also with defenders, and that means that it's a free first look, and if they don't like what they see, they don't have to call us again. So it's just a first look. Actually, it's interesting, prosecutors will often get a free first look. They don't like what they see. They turn it over to the defense, and then the defense might use that, but no, the science is the science, whatever it is. That has happened. And so we start with this data screening, and then we run it through the TrueAllele process, and then it's a very quick, we do a first look, and then we send back, at no cost, this preliminary report, and the report was answering his forensic questions and showed that there was information. And so at that point, because there was information on these other items, particularly items that included defendant, Lycoming then paid for a TrueAllele report. It's a 9-page report. In this case, there are a number of evidence items. This is just the first page, and this was the starting point of the evidence. You have a report, and now you can move forward. You have an expert, and either the other side accepts the report, or you start doing whatever lawyers do to go to court, have testimony, cross-examine, and so on. Okay, so these are the trial materials that we tend to start with. We have this report, and then we have a backup for every match statistic that we provide. So what our reports look like is we say, here are the evidence items, this is what we did, these are the methods we use, and we describe, this is how we analyzed each evidence item, we make comparisons, and then we provide match statistics. Numbers like 1 million if the item and a person are associated, or numbers like 1 in 1 million if they're not associated. It's whatever we're asked for. We don't tell people what they want to report. Sometimes prosecutors want to show that somebody isn't there. Sometimes defenders want to show somebody else is there. It's just science. What's the strength of the connection? And then we provide match statistics to different ethnic groups. We provide error rates, which I'll talk about briefly, which put these information measures into a context that jurors can understand. I'll describe that. We also, in addition to this case packet, provide a 4 gigabyte DVD. Nowadays, it's often by a Dropbox. And that has like 40 validation studies, 8 of them are peer-reviewed. It has court rulings from like 30 admissibility decisions. It has just many, maybe 100 different documents describing the TrueAllele process, how it works, the mathematics behind it. It also includes an invitation for the opposing side to test our software at no cost. Because in science, the real currency of whether something works is testing it. But we offer it. And when I'm involved in a case, if somebody has software, I'll run that software to see what their software will do, even if they don't even run their own software. I want to know what their methods do. but then I'm a scientist, so I care about that stuff. And as part of this, we also have PowerPoint slides that we use to educate the attorney, usually a prosecutor, could be a defender. Sometimes the police are in the room, they want to learn about it too. And then after they've done some background reading and we go through the PowerPoint slides, when we go to trial, this is what we show the jury. So I'm going to show you the exact slides that I presented in court. Cybergenetics has had a presence in Pennsylvania. We've worked on over 1,000 cases. There are 10 crime labs that use, in the US, that use our technology. In Pennsylvania, we've worked on over 200 cases. I think about half of them, the data came from the PSP crime lab. We've testified in 27 trials across 32 counties, which is almost half the state. And in the Foley case, which I talked about in 2014, that was 2009, and so we had appellate court precedent for TrueAllele's reliability in 2012. And we work in Pennsylvania mainly with the prosecution, but there are also defenders who've called us. And I'll mention that in some sense, all the cases we work on in our consulting work are cold cases, and that not enough has been done for the case yet, and more evidence is needed. And that evidence is a more rigorous computer interpretation of every possibility in an objective, unbiased, accurate way of the DNA evidence. The cases are waiting for the DNA to become informative. So I was asked to answer this question for you. When do you call the doctor, right? When is TrueAllele needed? And it's basically when the reported results you get back say that there's too little DNA. The words may be insufficient quantity. And that means based on the procedures that they follow or the software they use, whatever they're doing, they can't interpret it. They can't because maybe their tool could let them do it, but their protocols won't, or maybe they don't have a tool that lets them do it. So when it's a small amount of DNA, very few cells, you often get no answer. Sometimes it's the complexity of the DNA mixture that depends on the comfort level of a lab, its protocols, and what technology they use. There have been labs I've seen that wouldn't report a minor contributor to a two-person mixture, which to a computer is so easy, you wonder why you wouldn't do that. Some labs will set an arbitrary level of 3 people. They won't look at more than a 3-person mixture or a 4-person mixture. And these are arbitrary limits. What if it says there are no interpretable results? So that's their conclusion, because either there's too little DNA or there's too many contributors to a mixture, or it's too degraded. Sometimes you just see that the results are inconclusive. There are laboratories that don't use error rates, most don't yet, and so the match statistic, the likelihood ratio, will be below a level they're comfortable with. The statistic might be 5,000, but they have a cutoff of 10,000. They just won't report it. The evidence is there. Think of it, this can be particularly painful to people who didn't commit a crime, where there's evidence that supports that they weren't there and the lab just won't report it. So it works both ways. You have the DNA, it is informative, and the numbers aren't being reported for some reason. They're just called inconclusive. See that around the country as well. But TrueAllele doesn't have the limitations of other software. We spent 10 years building it before we released it commercially, and it learns everything from the data in that case. It doesn't need a threshold because it models the baseline with statistical variables at every level. It doesn't need to have calibrations done to figure out what the variation of PCR in a lab is, because it works it out on every sample directly from the data. On and on and on, whatever I'm saying, right? It just figures out what the evidence is trying to tell you without limitations, considering all the genotype possibilities. And the validation studies show that it's accurate. It's inherently unbiased because it can't use the answer that somebody might want because it doesn't know what you're going to compare with when it solves the problem. And with the error rates, there are no limits to what can be reported. we've reported maybe over 50 cases where the match statistics were under 200 because we report error rates. We give a context for a trier effect. Our methods are published. This is a paper from 3 years ago I wrote that is too mathy for most biologists. Don't bother, really, unless you like the stuff. But it writes what are the random variables, how does error work, how do error rates work, and their distributions for a single evidence item for validation studies. And this technology is in our software, which lets us calculate error rates in 1/100th of a second instead of taking 10 minutes to do it. So it's easy for us to do because it's built into our software. I think our current admissibility ruling count in the US is at 31. The first one on top is the Foley case from 2009 in Indiana County. And TrueAllele today, it's been out there for about 25 years. 10 labs use it. I think I've said all this before. We've testified over 100 times around the world and in the US. And of the 1,000 cases we've done, they've occurred in 45 of the 50 states, about 90% of the states. So it's widely used. And there's more information on our website, our YouTube page. If you just go to our homepage, cybergen.com, you'll see a lot of things there. And on our newsroom is actually a link to the World Trade Center.